Inherited immunodeficiency syndromes. Inherited immunodeficiency syndromes have recessive gene defects. The first is complement deficiency, that is characterized by recurrent infections by capsulated bacteria, such as Neisseria. Such capsulated bacteria avoid phagocytosis because these bacteria are engulfed only by phagocytes that see antibody or complement linked to bacteria. Second is Bruton's X-linked agammaglobulinemia. Persons with Bruton's X-linked agammaglobulinemia do not produce antibody. This pathology, the absence of antibody, is detected by immunoelectrophoresis. Such people are suffering from Streptococcus pneumonia. We know that immunoglobulins G antibodies are produced at 6 months after birth. Immunoglobulins M are immediately produced after the birth. If the person, the child, doesn't have immunoglobulins G at 6 months, then other child, healthy child, you can say that this is Bruton's X-linked agammaglobulinemia. Next is selective immunoglobulin A deficiency. Such persons suffer from chronic lung disease. Hyperimmunoglobulin M deficiency. Increased immunoglobulin M are unable to activate B cells, and there is a limited immunoglobulin M antibody responses against T cell dependent antigens. There is CD40 ligand molecular defect. We know that CD40 is critical in T cell dependent activation. They influence B cell proliferation. Such people with hyperimmunoglobulin M deficiency suffer from pneumocystis carinii, which is activated by macrophages. Then growth defects of complement components. Decreased activation of C3 component is characterized by pyogenic infections. Decreased activation of membrane attack complex C5 to 9 is characterized by infections caused by Neisseria, and decreased elimination of immune complexes, that is shown by autoimmune diseases. T cell immunodeficiency syndrome, DiGeorge syndrome. This syndrome is characterized by defect in thymic epithelium. T cells do not mature, and since T cell dependent antibody production is affected, cell mediated immunity is absent. We know that CD59 protects own cells from complement. as well as a decay accelerating factor. If there is defect in CD59, then paroxysmal nocturnal hemoglobinuria happens. And in this disease, red blood cells are destroyed. Next, we can explain C1 inhibitor defects. That is characterized by decreased activity of serine proteinase enzymes, as well as factor 12A and kallikrein. There is increased production of vasoactive mediators that results in fluid accumulation and hereditary angioneurotic edema. Two inherited immunodeficiency syndromes, chronic granulomatosis disease is also referred. In this disease, phagocytes do not produce superoxide radical, and chronic bacterial infections develop. In this disease, there is also defect of glucose 6-phosphate dehydrogenase and myeloperoxidase. Two inherited immunodeficiency syndromes, Chediak-Higashi syndrome is also referred. It is characterized by partial albinism, abnormal platelet function, severe immunodeficiency, and lysozymes do not fuse with phagocytes. Next is adenosine deaminase deficiency, when T cells do not develop. There are defects in interleukin 2, 4, 7. Then goes MHC2 class deficiency, when CD4 cannot be selected. There are complementing gene defects A, B, C, D, REXANC, RFX5, RFXAP, and two inherited immunodeficiency gene syndromes, Ullmann's syndrome is referred. This is similar to graft versus host disease that is characterized by rashes, eosinophilia, diarrhea, and also we can discuss such pathology as X-linked severe combined immunodeficiency, when there is a defective antibody response. Two inherited immunodeficiency syndromes, Wiskott-Aldrich syndrome is referred, when there is blood clotting disorder. Interferon gamma, interleukin 12 defects are characterized by accumulation of intracellular pathogens such as Mycobacteria, Salmonella. X-linked lymphoproliferative syndrome is characterized by defects in cytotoxic cells that leads to a Epstein-Barr viral infection. In all inherited immunodeficiency syndromes, bone marrow transplantation is prescribed. But bone marrow should be depleted the donor T cells in order to prevent graft versus host reaction.